This is the second half of Chapter 50, where we're going to move from how you sense something going on in the world to how you move to respond to it. This is a much shorter lecture. It's about half the length of the sensation one because it's really coming down to one thing. How do you flex a muscle? And that's what we're going to focus on now. Now, the arrangement of a muscle looks like this. You've got this big, thick thing called a muscle, and if it's on a cow, you call it a steak. Let's always remember that that's what steak or chicken is its muscle. Okay. Well, if you start breaking it down, I like to go bottom to top. There is a thing running through the length of a whole bunch of different cells. It's called a myofibril. It's a collection of heavy and thick and thin filaments. It's a collection of a whole bunch of cytoskeleton, basically. But it's cytoskeleton running in long, thin lines. So you've got myosin chains and you've got actin filaments. They're running the length of these. And they're bundled into cords. You know, there's lots of these things running through one cell. So I think of them as an organelle that can cut through many cells at once. So you go from the myofibril, which is kind of like an organelle. It's even wrapped in a membrane. You have many of these bundled up, okay, making a kind of a cable. And they're running through the center of a whole bunch of interconnected muscle cells. Okay, the muscle cells have nuclei as if they're individual cells, but they are not well divided from each other. This vertical line here is not really a full plasma membrane division, so there's a lot of shared communication between these cells. Okay, so this big horizontal thing is one cell with a whole bunch of myofibrils running through it, and these myofibrils are long enough to connect through multiple cells. You take multiple lines of these cells and bundle them together, and now you have the muscle, what we think of as a muscle, a bundle of muscle fibers. You take multiple bundles of muscle fibers, and then you have a muscle. Now, if we zoom in on one of these myofibrils and look really close at this organelle I've been talking about, we have a unit that we can consider called the sarcomere. Okay, and the sarcomere is one length of Z line to Z line. Now, why do we start and stop at the Z lines? Well, first off, they're very obvious under a microscope. But second of all, they are the anchor point that everything's going to be pulling against. Okay, so your actin filaments are attached at the Z line, okay, to basically one another. Okay, so it's a vertical connection of a whole bunch of horizontally spaced or vertically spaced actin filaments. Okay, so they all lock in there. Now, inside that sarcomere, you're also going to have an M line. It's a space where there is no overlap of the actin filaments. Okay, you also have some, they didn't really talk about these zones right here in the current text, so I won't talk about it here. There's also some spaces where there is no overlap of the myosin heavy filaments. Okay, now remember these little heads are going to grab hold of these actin chains and basically pull. And since they're pulling, one half of the myosin is pulling one way, the other half of the myosin is pulling in the exact opposite direction, the myosin is not going to move. What it's going to do is it's going to pull these ropes towards it. And it's going to end up overlapping these ropes and pulling the Z lines closer together. So that the size of this sarcomere is going to shrink because these Z lines aren't going to be quite so far apart anymore. Okay, and as they're pulled closer together, they're pulled closer here, but they're pulled closer along every sarcomere. So not only does not only are we pulling this way, we're also pulling on that side. So the net result of all that is all of this space in the middle here gets collapsed down. And that's how we're going to contract our sarcomere. It's called the sliding filament model. Here's your filament, an actin filament, and here's your myosin filament, and the two slide across one another to overlap more deeply. Okay, so that you've contracted the space. Notice that a muscle, when you contract it, gets thicker. Okay, because the cytoplasm, we're going to take this edge and push them both together. We're going to do that a lot. It's going to compress all of these cells together. Well, they can't go nowhere. They still have a defined volume. So when this line gets shorter, it also gets taller. So the size of this line right here, the diameter gets bigger. And that's what makes the muscle bulge. Okay, now this is what it looks like when you're contracting the sarcomere. You have that M line and the Z lines, and what happens is basically the M line starts to disappear. Okay, and so all that empty space there goes away. These Z lines were brought closer and closer together, and we have contracted the muscle now. The muscle got shorter. What's the purpose of making a muscle shorter? It's because if you pull this muscle shorter, whatever it's attached to has to come towards it. So when you flex a bicep, your bicep is connected up, around your shoulder, okay, and it's also connected to your forearm, okay, on the bottom half of your arm. And if you shorten that bicep, the forearm has to come closer. Okay, it has to get closer to your shoulder. Okay, it's not about your hand coming closer to your shoulder. It's about the point about an inch up from your elbow. So if you look at the inside of your elbow and you go 1 inch further towards your hand, okay, there is where your muscle is connecting. Okay, and you can even feel it if you flex it really hard. You can feel some tension in a ligament. A thing pops up underneath your finger. Okay, that's the connective tissue that's gripping hold of the bone. Okay, and as that thing pulls tighter, it's pulling kind of inward. Really, all you're doing is you're moving that 1 point, that inch out. You're moving it maybe 2 or 3 inches closer to the closer to the bicep. But that 2 or 3 inches down at the joint moves your hand a whole 1.5 feet as you flex it from straight to 90 degrees. Okay, and if that didn't make sense to you, I want you to write down a note, ask about that whole flexing the bicep demonstration, and I'll be able to point to my arm while I'm talking about it, and you'll go, oh, and you'll get it. But effectively, what would make a lot more sense, from a physics standpoint, is if your bicep attached out here at your wrist. Then when the muscle shortened, you'd get a very strong contraction. Okay, it would pull the whole hand that way. But that's not what it is. You know how when you open a door, you try to open a door at the handle and pull out at the very edge away from the hinge? That's because if you try to, if you tried to push a door open, let's say you were pushing a door open right at the hinge, it's very hard. You're moving that hinge like an inch, inch and a half, but the whole door is sweeping through like a 3-foot swing. Well, that's what we're doing with your arm. We're pulling at a hinge. We're pulling it closed by pulling at a hinge, and it's not necessarily the strongest way to pull, but it works well. All right, so how does this process work? Well, myosin is going to bind ATP and break the ATP. The phosphate's going to jump onto the myosin head, and when that happens, it's going to cause a change in the shape of myosin. Let's remind ourselves that's a tertiary structure change to where the head is going to reach outward. Okay, and that's the cocked position, and even when you're not flexing your muscles, they are in that cocked position, because the ATP is present, and then there they go. They're in that cocked open position. Well, after you get a signal that says flex, and we'll get into that in a minute, what happens is that head reaches up and grabs a little notch in the actin filament that's specifically designed for myosin to grab hold of it. Think of it as a knot on a rope, because if you're pulling on a rope, you want knots in it so that you have something to grip to. So once it reaches up and grabs hold of that actin, it releases the phosphate and the ADP that it was still holding on to. And by releasing the phosphate, it bends back to its original position. And notice the flexing here. We cocked outward when we bound the ATP. When we let go of the ATP, we're going to pull more towards this position. So what it does is it pulls that direction. Okay, but of course the myosin can't move because it's pulling on both sides. So instead the actin filament is pulled this way. Excuse me, in this scenario it's being pulled to the left. Okay, so we target it, grab it, and jerk over. Reach up, and then you do it again. Now this filament's been pulled somewhat. You put new ATP on here, cock the hammer again, reach up, grab, and pull yet again. And since we're doing this with thousands of little heads, it's not like when they let go, the filaments can slide back. Each time 100 let go, another 100 are still holding on in the middle of their stroke. And so it's a whole bunch of guys on a shoreline all pulling on a rope to try to pull a boat up. Okay, and that's the same scenario you've got here. The key to this process is if myosin can bind to these myosin binding sites, the muscle will flex. Okay, this muscle will contract. But if myosin cannot bind to those sites on actin, then the muscle will sit limp and won't do anything. Okay, here is where the regulation occurs. Something called tropomyosin, which is going to be a cord that is going to block those sites, and a control protein called troponin. Okay, so this is troponin, and this is tropomyosin. The tropomyosin is a cord blocking the myosin binding sites. Okay, you hear me, tropomyosin and myosin? Okay, it could get confusing. So the beads are actin. The little holes in the beads are the place where myosin is supposed to grab, and tropomyosin is blocking that spot. Troponin is this little rocker switch that can be regulated by calcium. If you dump calcium into the myofibril, okay, that's the unit we're talking about, that little organelle-sized thing. Okay, when the calcium gets in, it grabs hold into the troponin, and the troponin shifts, moves the tropomyosin out of the way, and now the binding sites on the actin are available. Now the regular old strong myosin can reach up, grab hold, and pull. And if you get rid of that calcium, then this troponin will roll back over, pulling this cable back across the binding sites and forcing myosin not to be able to interact anymore, and the muscle relaxes. So the key to this is how is it that calcium gets released into that myofibril? Because once calcium is released inside the body of the myofibril, you will get a contraction. A equals B in this case. And I'm a big fan of people distilling it down to the actual causal event of contraction. In this case, the causal event is calcium. Because if calcium is released, it immediately binds to the troponin, moving the tropomyosin out of the way, and we get flexation. That's your control point. The control point is not a nervous signal. The nervous signal will simply signal the calcium. But the calcium is really what's in control here. Okay, so how do we do this? Well, let's start with regular neuron. Action potential, action potential, action potential. So this wave of sodium rushing in and depolarizing our cell causes more sodium to rush in, more depolarization, until you get down to the synaptic terminal, down to the synapse. And instead of sodium channels opening up, now it's calcium channels that open up. Wait for it. That's not this calcium. Okay, that's not here. This is in a neuron. This calcium gets used for many things. So calcium rushes in, causes vesicles at the end of your synapse, okay, to fuse. Remember from chapter 48, when calcium channels open up and calcium flows in, vesicles suddenly fuse, just like they did in the slow block to polyspermy back in chapter 47. Okay, when an egg gets contacted by a sperm, calcium caused those vesicles, those cortical granules, to fuse with the plasma membrane and dump their contents. Okay, so calcium rushes in. These vesicles fuse, dump out acetylcholine, which binds to some ligand-gated ion channels on the surface of your muscle cell. Okay, so this is on a muscle cell. This is on the outside of the bundle, and there are many myofibrils inside that muscle cell. Okay, and so what happens next? Well, these channels that open up are going to allow in some sodium. Okay, and we're going to, now we've depolarized the muscle cell, and the muscle cell will act just like a neuron at this point, in that it will have its own action potential fire. But instead of firing it down a long axon, what it fires is along its own body. Okay, I mean, that's what's happening in a neuron. This axon is just an elongation of its own body. Well, the muscle cell fires it along its own body, including it cuts that same wave of charge, and depolarization cuts its way down into these things called T-tubules, which take you into the center of the cell. Okay, which happens to be where all these myofibrils are. So now that this action potential's come down to this area, as it's going through, it passes by a specialized endoplasmic reticulum that's been wrapped around these myofibrils. Okay, you can see it here around each of them. It's called the sarcoplasmic reticulum, for sarcomere. And as the action potential passes down, as you get these depolarizing events coming through, suddenly voltage-gated calcium channels open. Okay, so when depolarization happens to the sarcoplasmic reticulum, voltage-gated calcium channels open up, and they allow calcium to dump out. And now the calcium binds to the troponin, and the troponin rolls sideways, pulling the tropomyosin out of the way, allowing the myosin heavy chains to grab hold. So, let's talk about this from the perspective of rigor mortis. Okay, so a person dies, and to begin with, their muscles are all flexible because there's no nervous signals telling their muscles to do anything. But then, eventually, the calcium, the sarcoplasmic reticulum, breaks down. And then all of these, all the calcium dumps out, and all of the muscles start to try to contract. So part of that starts to, tense up the body. It starts to make it get tenser. Further, the body starts to run out of ATP. Okay, while we had ATP, we could be cocked and ready to go. ATP is actually what made us let go of the actin filaments. If the body runs out of ATP, these muscles eventually run out. Okay, then what happens is now this thing is stuck grabbing hold of the actin. Okay, so now it's grabbed the actin and it can't pull anymore. It's out of ATP, but it also can't let go. So that's the other thing that happens with rigor mortis, is not only do the muscles start to kind of contract a little bit, but the muscles also become locked in place, and it becomes very hard to move the person's arm because you have to physically break all of these myosin head connections to the actin filaments. And when you break them, when you let go, then they tend to kind of reform wherever they were at. Because the purpose of the ATP is not to allow it to make the connection. The purpose of the ATP, okay, is that it causes it to be cocked outward and able to let go. Okay, ATP helps it let go. Breaking the ATP cocks it outward. It can grab hold without ATP all it wants to, and that's where your rigor mortis comes from. Okay, these are just words that say everything I just said. Now, we know that action potential is one and only one thing. It is what it is. So how is it that a muscle can flex halfway if all it's receiving is action potential, one signal? Well, the way is you can vary the number of fibers that contract or how quickly they contract. Okay, a motor unit is a set of muscle fibers. Okay, so here's the muscle fibers with their myofibrils inside them. These muscle fibers, remember, are one cell thick, right, but many cells long. Multiple of these muscle fibers will connect to the same neuron coming out of your spinal cord, for example. Others of them will be connected to a different neuron. Okay, so they don't all contract at the same time necessarily. If you're just trying to move your hand about halfway up, maybe it only fires motor unit 1 but not motor unit 2, so only these contract but these do not. And these guys are not strong enough to pull the arm all the way up, they only pull it halfway. But if we fire all of these at the same time, now we're going to contract the entire arm. Just contracting your arm never takes all of your motor units. Your arm doesn't weigh very much, but what about when you're doing like curls with a big heavy weight? I love working with the bicep because it's very obvious and it bulges when you contract it, and you can see the simple physics of it pulling straight. Well, if you're trying to lift something that's much heavier than the actual arm itself, clearly more motor units are going to have to get involved to lift that weight. When you're in training, when you're doing sports medicine, they start talking about different types of strength. They'll talk about raw strength, but then they also talk about a concept called power. Power is the ability to explosively exert your strength. So it's the idea not only to just curl that dumbbell up in your bicep, but to snap it up as quickly as possible. See, two people might be able to lift the same dumbbell, but one can lift it, much quicker than the other. A very strong bicep curl does not correlate to a very strong uppercut punch. It means you could press your fist harder into something, but it doesn't necessarily mean that you'll hit it more rapidly. So why am I talking about power all of a sudden? Well, see, strength is measured based on the thickness of the muscle fibers. You cannot add more of these muscle fibers necessarily, but what you can do is add more units of more sarcomere thickness in there, basically. You can add more of the, let me get the word here, I want to be able to point to it. What you can do is you can add more of the myofibrils inside each muscle fiber. So you don't add more cells, you make each cell thicker by adding more strands of actin and myosin to be pulled on. That is raw strength, and that's how much weight total you can pull. Power is how quickly you can recruit all of those now much thicker muscles in order to make a motion. Okay, so one of the interesting things about this is people talk about, a mother whose child is trapped and underneath this gigantic heavy telephone pole, and the adrenaline causes her to have super strength, and she lifts this gigantic heavy telephone pole off. How can that be? And people assume the adrenaline is somehow an energy source that powers the muscles and supercharges them. That's not it at all. Adrenaline takes controls off. Adrenaline says, you know what? Screw it. Every motor unit flex now. And the motor units say, we've not practiced at that. And it says, I don't care, you're flexing. Now, go. And every motor unit flexes. Whereas before, that mother had controls on her biceps that said, you know what? If we're flexing all of these motor units, but the arm isn't moving, we're not going to try to flex anymore, because if the arm isn't getting shorter, if that muscle isn't actually allowed to contract, what we're really doing is we're pulling harder and harder, and we're going to tear the muscle. So adrenaline comes in and says, yeah, no, not worrying about that. And so what they tell you about is the fact that the mother lifted the gigantic heavy thing off her car. What they don't tell you is that she spent 3 days in the hospital largely unable to move her arms afterwards. Once the adrenaline wears off and she's realized she's put muscle tears in 6 different muscle groups in her, upper torso and arms, and in her legs, because let's not forget that her legs are supporting the weight too. Okay, it's the sudden willingness to exceed the strength of the attachment to the bone and the muscle itself. Okay, so people are a lot stronger than they think they are. What they are not normally used to doing is exerting it, and really exerting their full and true strength. And that's one of the things that gets trained into you when you're in athletic training, is how to recruit extra motor units and how to recruit them faster. You've heard people say that that guy's not strong, but he's wiry. Okay, that kind of doesn't make much sense. He doesn't have stronger muscles, but he looks like he can do more than other people can. It's just because the person has practiced not strengthening the fibers, but recruiting the fibers, has practiced that power concept, has practiced, thinking hard every time he does that really hard pull on the arms, thinking, I want to pull as hard as I can, not just hard enough to lift this thing, but way harder than is necessary to lift this thing. He doesn't gradually recruit them, he recruits them all at the same time. Okay, and that's just a little bit of functional understanding of what a motor unit does. Okay, now we can talk about fast and slow. Now there's different types of muscle fibers here. We can talk about oxidative versus glycolytic. Okay, oxidative are the ones that are going to rely primarily on aerobic respiration. They're going to have lots of blood vessels. They're going to have lots of mitochondria. Okay, myoglobin is kind of like hemoglobin. Myoglobin is hemoglobin that does not circulate. It traps the oxygen right there in the muscle to dump it for just that muscle. Okay, so it's just like hemoglobin in its ability to grab oxygen, but it keeps it inside the muscle cell and does not let it go anywhere. It doesn't let it diffuse. Okay. Your oxidative fibers look like that. Lots of mitochondria, lots of blood supply, and lots of myoglobin. Your glycolytic fibers, okay, do not. Okay, they use glycolysis primarily. They have fewer mitochondria. They don't worry so much about blood flow because they're not going to need the oxygen. Okay, they are things that do lots of anaerobic work. Okay, so these things tire easily, but they usually can give you more instantaneous power. Okay, they're designed to be a bit faster. Okay, so when you're talking about light meat versus dark meat, okay, light meat is the glycolytic. Okay, the dark meat is the long distance stuff. Okay, it's the oxidative. All right, so birds are used to their the wings being something that has to move a lot constantly, over and over, and so that's why the wings tend to be, and the legs tend to be that dark oxidative fibers, is because they're not things that are going to do, that are going to just snap into motion. Okay, this comes down to the idea of slow twitch and fast twitch. Okay, so slow twitch fibers are the oxidative ones. Okay, they are not for rapid reflex; they are for jogging. Okay, so the slow twitch fibers, which are oxidative, are going to be dark meat, for example. The fast twitch fibers, which are going to be for rapid bursts of speed, sprinting. Okay, they can either be glycolytic or oxidative, but they've got a mix of the two usually. Okay, so they've got more glycolytic in there. With your slow twitch, it's always oxidative, but with fast twitch, they introduce some of these glycolytic ones. Your muscles are made of both. Yours are. Okay, and birds are weird in that they separate the different types of fibers, and that you have this, glycolytic breast meat on certain chickens. I got to tell you, that's also a bred characteristic. We've bred that into them. A wilder bird would have a more even mixture of meat colors, would not have that pure white breast that chicken seems to have. It also would not be nearly as large. Okay. Now, so that was strength versus power, loosely correlating to motor units that you recruit, but also what type of fibers you've got, oxidative versus glycolytic. If you're looking for raw strength, you're looking for glycolytic. If you're training in aerobics, you're adding more oxidative. Okay, that's also a thing that is affected by muscle workouts and how you work out. And then you've got your glycolytic ones that can be fast twitch versus your oxidative ones, which tend to be slow twitch but can also be fast twitch. And these get you different kinds of motions. Right now I'm bouncing my knee up and down. Okay, I'm using fast twitch because it's rapid back and forth, back and forth, back and forth. My throat just then, my tongue were doing fast twitch because I was going back and forth, back and forth, back and forth. That was rapid motions. But slow twitch is like when I move my leg and cross my legs. That's a lot of slow twitch fibers working together. Other types of muscle, you have cardiac muscle. Okay, it's cross-connected. These guys don't work in straight lines. They work more like a net. They still pull, okay, just like all muscle fibers pull. Okay, but what they do is they collapse down a volumetric space. They are involved in volumetric contractions. So instead of pulling in straight lines, they pull like they're pulling down the sides of a net. Okay, so they do pull in straight lines still, but then the lines change angle. Okay, and that's what that intercalated disc thing is. Instead of a whole bunch of straight, instead of parallel pulling, they pull at odd angles from each other, which causes, instead of a linear contraction, it causes a volume to contract. And that's good. It pumps your heart. Also, cardiac muscles generate their own action potentials kind of to begin with. They're that primed to pump. Okay, and that's how the heart can sometimes restart even in the middle of a heart attack. Smooth muscle. Okay, these are like your digestive tract. These guys do linear contractions in a radius. So really it's a radial contraction. Instead of contracting, taking a length of small intestine, let's say it's a foot long, instead of shortening the length of the small intestine, what these do is these myofibrils wrap around that tube, and as they contract, they squeeze the tube tighter. So skeletal muscle was a linear contraction. Cardiac muscle was a volumetric contraction, making the volume smaller. Smooth muscle is a radial contraction, making the radius smaller, tightening the tube. Okay, I'm not going to go through all the parts of the skeleton. I don't think we need to. Think we need to talk about all the types of skeleton. Your brain's almost full. What I want to talk about is the basic scenario of what a skeleton is there for. A skeleton is not protection. Yes, it protects your brain. But here's the key. A skeleton, either endo or exo, is absolutely necessary in order to push. Okay? Because muscles cannot push. And yet you can do a push-up, right? You can lay flat on your chest and press your arms outward from your body. A squid cannot do that. If you ever see the little cartoon shows with a squid jumps up on land and starts running on its tentacles, it cannot possibly do that because it cannot push its weight up because it has no skeleton. It can pull. It can constrict you to death with those tentacles. It can wrap them around things and tighten. It can do the linear pulling, but when it comes time to relax that, it cannot push the tentacle back outward. All it can do is let the tentacle dangle. It can relax it. The tentacle will slowly extend on its own. An octopus could never backhand you. Okay, it cannot slap you. Okay, it can squeeze you to death. That's what it does. Okay, so how is it that you push? You don't. You pull on bones. Here's how push-ups work. Push-ups do not help your bicep any. What they help is your tricep. Okay, they make this muscle bigger. And how do they make that muscle bigger? Okay, they are a flexor muscle. The biceps is a flexor muscle. It can pull your arm upward. In the process, your triceps has to thin out because it's attached to the exact opposite side of the same bone. So while the connection between this muscle and this bone is shortened, it's pulled inward because the muscle got shorter, so it pulls that connection point inward. This muscle has to thin out because this line was just lengthened. But if you want to press your arm outward, if your arm is bent, not just curled up against you, but let's say it's bent and pressed against the ground, if you try to collapse the triceps and bulge it up thickly, what it's doing is its attachment point is being pulled around this direction because going around the curve is longer, and if you pull these two bones into a straight alignment, this distance is now shorter. Going around a curve is longer than the shortest distance between 2 points is a straight line. Well, if you pull these two bones into a straight line, then suddenly this muscle can be pulled tighter. And that's what's happening. It's not that you're pushing your arm, you're pulling your arm back into a straight alignment, and it just so happens that shoves upward and presses your whole body up, and you do this amazing thing called a push-up. It's the same thing when you punch. In fact, punching someone does not rely on the strength of your bicep. Okay, pulling someone would rely on the strength of your bicep. punching someone is more of a tricep motion because it is snapping straight the arms and these two bones. Okay, so muscles always work in pairs for that reason. They need bones to pull against, whether it's pulling in an external bone or it's pulling on an internal bone. It's got to have a bone to do it. Okay. You can read these. They're just some simple statements about efficiency and the concept of here. I'll tell you this one. If you're swimming, you expend less energy than if you're running because you're not resisting gravity when you're swimming. Okay, and if you're trying to travel, it's very much more energy efficient to be big than it is to be small. Though small things are very much more efficient at changing direction rapidly. But if we're going for a long distance run, a big thing going for a long distance run burns fewer calories per kilogram or per gram than a small thing. So a mouse has to burn a lot more energy to run the same distance per gram of mouse than you do. Okay. So if we took a whole bunch of mice that weighed the same thing that you did, I know that's a gross idea, but work with me here, and had them all walk across the room and measure the energy they expended, moving the exact same weight that you weigh, and then we measure the energy you expend as you walk across the room, we find out you expended less energy than they did to move the same mass.